cyclin D. Oh, sorry, it's CDK46. Okay, I can tell you. Okay, this is active. Okay, I'm going to show you another substrate, and I'm going to deviate a little bit from my notes because I thought, this is got to be an easier way to make things look less confusing. Okay. So CDK4/6 also can work in the nucleus, it works in the cytoplasm, but in the nucleus, the main target you know what, just to make it easier, I'm only going to draw one E2F, okay? There's usually, there's going to be two, but let me just make life a little easier for everybody. Let's only put one in. Okay. E2F's not the target. The target is that protein. Oh, and let's do it in, I'll put it in different color. This is the retinoblastoma protein. It was discovered in the cancer's retinoblastoma, which is it's actually a childhood cancer that appears early, like 1 year old, in one eye. So a tumor starts forming in the eye, and you can kind of tell a milkiness about the pupil. Usually it involves both eyes. Yeah, perhaps.
Would that be on, so you said usually there's two. Is that on just one of them, or is there one on both?
Oh, you know, I don't know, because in the heart, no models will show you, you know, what's really going on. All models are sort of, this is what's going on. I don't know the stoichiometry. So, in other words, so it's not important. Retinoblastoma found. In my old notes, I had retinoblastoma and the transcription factor floating around in the cytoplasm, but that's not what's going on. And I thought, oh, you guys just can't figure, you know, you're not smart enough for this. But yeah, you are, okay? You're smart enough. E2F is sitting on its particular enhancer region that it recognizes. Retinoblastoma protein is bound to it, and there's no transcription, okay? And for those in molecular genetics, it's really because, okay, there is a protein that you guys know. And I don't know if it's bound to A2F. I think it is not. It is not. So let's say histone deacetylase, and only bind it to. Okay. So here's one of the things. So it's not just also that retinoblastoma is bound. It's that there's also a histone deacetylase. Now, if you remember back from 111, back in the beginning bio, when you acetylate histones, you can turn on genes. In fact, it's a requirement for gene activation is that you acetylate the histones in the promoter region of a gene. Okay? If you deacetylate the histones, okay, and HDACs remove acetyl groups from histones, you turn off the gene. This is not permanent. You can kind of go waver back and forth. All you have to do is get another enzyme to acetylate the histones. But in this case, the genes that E2F control are turned off. Okay? Partly because retinoblastoma protein is bound to the transcription factor, and partly because it's recruited histone deacetylase to take any acetyl groups off the histones. Okay? So some people are probably like, oh, of course. Okay, so that means no gene transcription. Okay. All cells rely on retinoblastoma, so even though it's called the retinoblastoma and its mutation or silencing, it's usually both a mutation and a silencing of the gene, always is associated with retinoblastoma. So it's kind of odd that some proteins are associated with particular cancers, even though that protein functions in all cells of the body. This happened to be the very first tumor suppressor gene ever discovered, or tumor suppressor protein ever discovered. Prior to that, I don't think people were really looking for proteins that block tumor formation, but this was the first one that to be discovered that actually is associated with blocking tissue formation. Okay, so here's what cyclin D, CDK246, that's cyclin D, CDK4/6. Okay, when it is active, it's going to phosphorylate the retinoblastoma protein at the position where the histone deacetylases bind. So let's redraw this. 2F is still there. Retinoblastoma protein is still there. Okay, the two phosphate groups at the histone HDAC site okay, but the phosphorylation bumps the histone deacetylase off, and that allows now genes to be turned on because now the histones can be acetylated by acetylating enzymes, but not deacetylated. Okay, so what we're going to see then, with the loss of the acetylating enzyme, now we can see genes being turned on. Okay? So let me just to make sure, because it's really fresh in our heads because we just had a test in molecular genetics. Removes acetylating SC groups. Okay, and so I'm going to step back a little bit just because to give you guys some perspective. Okay, so histones that don't have an acetyl group, okay, this is a lysine. Histones are rich in lysines. This is the R group for lysines. And I'm going to draw another lysine. Oh, there's a ton of lysines in histone. This is lysine. Single abbreviation letter is K. Okay, and the acetyl group is, okay, that, I don't know if that's showing up very well. That probably doesn't show up very well on the videotape at all. But notice we lost the charge. So when when histones are not acetylated, a very tight bond forms between, well, not tight, it's an ionic bond forms between the lysine and the histone. Okay? So the histone has a grip on the DNA. And that means if the histone has a grip on the DNA, no other proteins can access the DNA. So RNA polymerase II can't transcribe the gene if there's a tight hold on the DNA. But when you acetylate the lysine, removes the positive charge on the nitrogen, on the amine group, and now that frees the DNA from the histone. Now it's easier for other proteins to gain access to those, to the DNA. RNA polymerase, for example, might be able to bind if the histones aren't grabbed onto the DNA. So acetylation always loosens up the chromatin, loosens up the histones and the DNA, so other proteins can bind. Okay, so that's like the nickel version of what acetylations do, but it's good enough. So the removal of the acetyl groups, in other words, going back to this, causes the histones to be tightly holding onto the DNA so other proteins can't have access to the DNA. Okay, does that make sense how histone deacetylases, the removal of the acetyl groups, closes up the DNA so the histones have a tight hold on the DNA and RNA polymerase doesn't have access to the DNA anymore, cannot transcribe the gene. Okay, does that make sense? I mean, it makes sense to everybody who's had molecular genetics or currently in molecular genetics, but if you haven't, how does that sound? Okay, it's sort of the version we cover in Bio 111, but we expand the role of acetyl groups a little bit more later. Okay, so let's take a look at what gets turned on. Cyclin E. So that's how we get cyclin E. Okay, first we need to cyclin D CDK4/6, okay, to change the protein arrangement at the promoter region. And then we can get cyclin E to be transcribed and translated. Okay, we also get P16, okay, remember the CDK inhibitor that says, okay, there's enough of us, we don't need to divide anymore. Okay, that's the one that's going to end the cell cycling. And the other protein from the same gene, ARP. Okay, probably see ARP roll tomorrow. Okay, maybe tomorrow. Okay. CDC25. Remember, that's going to remove the inhibitory phosphates on the CDK2. Okay. So we're going, and right now that's it. Okay. We still need to change the retinoblastoma a little bit more. Okay. It's not fully. It has not fully activated E2F. It's activated E2F to make at least some of the proteins it can activate, but not fully. Okay. I'm going to skip a little bit here. So once we get cyclin E, I think I did this in the wrong order. CDK2. Okay. So what we needed to do is we needed the CDC 25 in order to get rid of these inhibitory phosphates. Activated CDK2, okay? So we turned on the gene for CDC25. CDC25 can remove the inhibitory phosphates off of CDK2. That activates CDK2. Okay? And CDK2 is also going to phosphorylate the retinoblastoma protein. Okay, so this is what the next picture looks like. Okay, the retinoblastoma protein will release from the E2F. Okay, of course, this histone gets settling, you can't find. And that allows E2F to be fully functional. Okay. And let me know if I go too fast. Are you ready for what it now is going to transcribe? Cyclin A. Okay? So do you remember we saw in the very first day of this unit, first we see cyclin D appear, okay? And that's dependent on the growth factor. And then after cyclin D appears, cyclin E appears. Okay? And then after cyclin E appears, cyclin A appears. Okay? I've never used this picture before. I've always said, well, cyclin A appears in a cyclin E dependent manner, and so, but this is the first year I said, oh, this is actually how cyclin E appears. And I'm also sort of lying to you because you're getting the nickel version of histone modifications, because there's, for those of you who are currently in cell, there's some SW1 SNF involved in getting activated by cyclin E. CDK2, but I don't think we need to do this in this course. Okay, any questions about the sequential gene activation by E2F?
I thought SNF only involved when you have histones.
SW1 SNF?
Yeah.
Yeah, so from going to here, so one of the things that's activated here is SW1 SNF.
But there's no histones right there. This
Oh.
is-
Well, there would be, like here and here. We didn't get rid of all of them, they're still there. Remember how in gene activation, always an acetylation, bunch of acetylations required, and always chromatin remodeling by SW1 SNF. So some of the features that all seem to be here for all gene activations is bumping the histones out of the way and acetylating them. But not that way. Yeah, right.
Does it still transcribe for
Oh,
cyclin E?
You still make E and
Yeah.
still make A? Yeah, I don't know for how long, but you'll notice that if you look at, and also you can see this in your textbook, there's overlap of where you find cyclin E and cyclin A. They're going to appear, they're going to be present at the same time for part of the cell cycle. Okay, so let's see, what have we got so far? If we go back to what are the three things that we still have to accomplish? We've got cyclin E present, okay, and the cyclin, and it required cyclin D. We got cyclin A present, so we got the cyclins that activate CDK2. We turned on the gene for CDC25 to make the phosphatase that removes the inhibitory phosphate. So we took care of those two ways of activating CDK2. What's the third thing we've got to do? Got to get rid of P27, the thing that is holding CDK2 hostage. Okay, so this is going to be, and there are three strategies. this one. Who's gonna be, here's what we need to do. E and A, we did that. Removal of inhibitory phosphates. Okay, got that taken care of. The activating phosphate is just gonna happen because this kinase that the CAC is ubiquitously present. It is ubiquitously present, and it's always present in the nucleus, okay? So now we have to do inactivate P27. Okay? So I'm going to list the 3 strategies. That's not strategies I want to talk about. Of inhibiting P27. Well, we can do it. One is to lock P27 in the cytoplasm, okay? Because you're going to find CDK2 in the nucleus. So if you keep P27 in the cytoplasm, when CDK2 is in the nucleus, you can't inhibit, P27 can't inhibit CDK2. Okay, that's one. You can ubiquitinate and destroy it. Okay, and that's ultimately what we're going to see happen. And you can sequester it, P27, on CDK4 or 6. Okay? So what this means is that if P27, let's give me P27, is bound to CDK4/6, it doesn't really inactivate CDK4/6. You can have cyclin D on there and that complex is still active, but what we see happen is that if P27 is bound to CDK4 and 6, then it's not available to bind to CDK2. So that's just another strategy, just sequester it by binding it to some other protein. Okay? You can silence, and then in a cancer cell, what we see very rarely, as a matter of fact, is the silencing of the P27 gene. That is, of all the tumor suppressors, this is probably one that's least silenced by DNA methylation. Okay, so here is it's a long pathway. You're going to need a whole page. Sorry, but you're going to find you're now going to find out some viciousness of AKT. And by the way, just to prime you, this cytoplasmic location often gives a very poor cancer prognosis. Okay, so that's what I wanted to take a look at. What would you need to do to lock P27 in the cytoplasm permanently or semi-permanently? Because even though P27's gene is not mutated or silenced, what they see oftentimes in cancer cells is P27 is present in the cancer cell, but it's stuck in the cytoplasm where it cannot do its job. Okay, so we're going to need to involve some by the way, this is, if you can't recognize it, this is PIP3. Okay? Sorry about that, it's kind of cruddy PIP3, but that's what that is. Okay. And as you know from the last test, you have to have PIP3 as a docking site for PDK and AKT, so PDK can activate AKT by phosphorylation. And one of the substrates, is P27. And the phosphate group is put on T157. Okay. Looks good. Okay. Okay. So here's the problem with this phosphate group. It's not gonna. It's not in a good location. The phosphate group is located in the address label called the nuclear localization signal. Okay, so what that means is that the address label is rendered inactive. So the protein that would recognize the nuclear localization signal, kind of that escort protein that would bind to P27 and escort it to the nucleus, doesn't recognize the phosphorylated and nuclear localization signal. So what's going to happen to P27? It's going to stay in the cytoplasm. Okay, so that means P27 remains in the cytoplasm. Okay, so at the end of last unit, we noticed that one of the overall activities of PAKT, when it phosphorylates a bunch of substrates, is to promote cell cycling and cell survival. Okay, so here's one of the ways that active AKT can promote cell cycling. Keep P27 away from CDK2. Okay, so that is a bummer. Can you imagine then what's going to happen to the location of P27 if you knock out P10 activity? How's that gonna shift the location of P27? It's gonna be almost constitutively in the cytoplasm. You're not gonna be able to move it into the nucleus if we maintain PIP3 and not break down PIP3 to PIP2. This is a better picture. Okay. So this is one way. Okay? And again, this could also, so this is going to be way number 1. Or it doesn't match that. We're going to just keep P27 in the cytoplasm. Don't let it move into the nucleus where it's actually going to have some effect on CDK2. The other thing you can do at this point is, and this is, let's let it get into the nucleus. Okay, so here's the cytoplasm. If it doesn't get phosphorylated by AKT, it'll probably migrate into the nucleus. So let's have it go into the nucleus. Okay, where it could become sequestered by CDK4/6. Oops. Didn't use enough space for that. So this is option number 2. All right, just bind the P27 to CDK4. Okay, so that it is not available to bind to CDK2. And I did not put cyclin D in there because I don't think that cyclin D is an essential is essential for the binding of P27 to CDK4. Okay, so here's another option. This option is going to take place after, if there is a functional CDK4 or 6, okay? So let's say, when the question is there a reason for me to divide? And the answer is yes,